Today's topic is ion channels in plants, and in this class we'll be discussing plasma membrane ion channels and vacuolar membrane ion channels. Most channel proteins in the plasma membrane of animal and plant cells that connect the cytosol to the cell exterior necessarily have narrow, highly selective pores that can open and close. Because these proteins are concerned specifically with inorganic ion transport, they are referred to as ion channels. So, an ion channel can be defined as a protein structure embedded in the cell membrane that acts as a pore allowing the passage of specific ions across the membrane. These channels cannot be coupled to an energy source to perform active transport. So the transport that they mediate is always passive, means downhill. Thus the function of ion channels is to allow specific inorganic ions, primarily sodium, potassium, calcium, and chloride, to diffuse rapidly down their electrochemical gradients across the lipid bilayer. There are two important properties that distinguish ion channels from simple aqueous pores. First is they show ion selectivity, permitting some inorganic ions to pass but not others. The second important distinction is that ion channels are not continuously open. Instead, they are gated, which allows them to open briefly and then close again. In most cases, the gate opens in response to specific stimulus. The main type of stimuli that are known to cause ion channels to open are first is change in the voltage across the membrane, and the channels are called voltage gated channels. Second, mechanical stress for mechanically gated channels, and third, the binding of a ligand, and the channels are called ligand gated channels. More than 100 types of ion channels have been described thus far, and new ones are still being added to the list. They are responsible for electrical excitability of muscle cells in animals, and they also mediate most forms of electrical signaling in nervous system. In plants, for example, they propagate the leaf closing response, as we have studied in Mimosa pudica plant. In this class, we'll be discussing only ion channels in plants. Since the first recordings of single potassium channel activities in the plasma membrane of guard cells more than 25 years ago, Patch-Clamp studies discovered a variety of ion channels in all cell types and plant species. Mature plant cells. Just look at this figure, first part of this figure, this one. Mature plant cell. This is a mature plant cell. They are equipped with chloroplast, a large central vacuole, and cell wall matrix, uh, enclosing the plasma membrane. So many other organelles are also present in the plant cell, like mitochondria, Golgi apparatus, endoplasmic reticulum. But here only chloroplast, a central vacuole, and cell wall is shown because they are only present in the plant cell. Due to the difference in osmotic pressure, low in extracellular space but high in the vacuole lumen. Here, because of high concentration of stored osmolytes, a darker pressure pushes the thin cytoplasmic layer against the cell wall. The cytoplasm, this part, is sandwiched between two extra cytoplasmic compartments. So, solutes taken up from the cell wall space often end up in the vacuole. Now just look at the second part of this figure, this lower part. This part is showing plasma membrane. This is plasma membrane and vacuolar membrane. So above the plasma membrane, this part is apoplast, this part is cytoplasm, and the lower part, this is vacuole. So this plasma membrane and vacuolar membrane, both are equipped with an individual set of cation anion channels. Just look at this part. These are cation and anion channels, carriers, this is carrier, and pumps to provide for solute and ion exchange with both extracytoplasmic compartment, that is apoplast, and vacuole. Specific proton pumps in both membranes are, just look at this part, PATPs in the plasma membrane and VATPs and PPIAs in the vacuolar membrane. They acidify the cell wall space and vacuolar lumen and polarize the membranes. So first of all, we'll start with the plasma membrane ion channels. Here we'll study the cation channels and anion channels. In 2000, the genome of Arabidopsis thaliana became available. The number of potassium channels increased to 15. 9 shaker channels, 5 two-port K channels, and a single IRK-like K channels. So among them, all shaker-like channels could be localized to the plasma membrane. So first we'll discuss shaker potassium channels. Shaker potassium channels are a type of voltage-gated potassium channels, and they are characterized by their 6 transmembrane domain S1 to S6, and they play a crucial role in regulating potassium ion flux across the membrane. So the examples of shaker potassium channels are SQR and GORK. GORK is expressed in guard cells and root hair, where it operates as an outward rectifying potassium selective channels. Then SKOR is localized in the vasculature, namely the xylem parenchyma cell, where it is engaged in the solute loading with the xylem network. SQR, as well as animal shaker channels KAT1 and KAT2, are activated by hyperpolarizing potential and therefore operate as strict inward rectifiers. KAT1 and KAT2 are predominantly expressed in the guard cells and the phloem network. EKT1 is primarily found in the plasma membrane of plant roots and play a crucial role in potassium uptake from the soil. AK2 is expressed in the phloem network, a low resistant solute pipeline of interconnected non-vacuolated and non-nucleated cells. Next is calcium channels. Plant cells utilize various calcium channels in their plasma membrane to regulate calcium influx and efflux. So we know calcium is a central player in plant signal transduction. Different types of calcium channels are number one, voltage gated calcium channels. These channels open in response to change in electrical potential across the plasma membrane. So depolarization typically activate these channels, leading to calcium ion influx. Second is hyperpolarization activated calcium channels. These channels open when the membrane potential become more negative, and they are less common than voltage gated channels but play a role in certain cellular processes. Next is voltage insensitive calcium channels. So these channels open and close independently of the membrane potential. And next type is mechanosensitive calcium channels. These channels open in response to mechanical stress or change in the cell shape. And next is ligand-gated calcium channels. These channels open when specific molecules, means ligands, bind to them. Example include cyclic nucleotide-gated channels and glutamate receptor-like receptors. Then two-pore channels, TPCs. These channels are located on both the plasma membrane and endomembranes and are involved in the calcium signaling pathways. Next is plasma membrane anion channels. In plants, plasma membrane anion channels are primarily categorized into 3 types based on their activation properties: depolarization activated, hyperpolarization activated, and those activated by light or membrane stretch. So depolarization activated channels are further divided into R type, means rapid activating, and S type, slow activating channels. These channels play a crucial role in various plant processes, including stomatal movement, anion homeostasis, and salt tolerance. R type means uh, rapid activating channels. These channels they activate quickly upon member depolarization, and they are found in various plant species and tissues such as guard cells and root epidermal cells. S-type, slow activating. These channels activate more slowly than R-type channels upon depolarization. And now, different examples of rapid activating and slow activating channels. First is SLAC1, a well-characterized S-type anion channel in Arabidopsis guard cells, and this is involved in stomatal closure. Second type is ALMT12 and oblique QUAC1, a rapid type, means R-type anion channel in Arabidopsis guard cell, and they are also involved in stomatal closure. Next is ARAC, Arabidopsis root anion channel, depolarization activated anion efflux channel found in Arabidopsis root, epidermal, and root hair cells. And the last one is VMAL, vacuolar mallet channel, and anion channel in the tonoblast. Tonoblast means vacuolar membrane of CAM plants involved in uh, malate transport for diurnal regulation of carbon metabolism. Next is vacuolar membrane ion channels. Vacuolar membrane is equipped with transport proteins that mediate the passage of a wide spectrum of compounds. Among them, ion channels, H pumps, and H coupled carriers have been shown to represent major membrane conductance. Transport processes across the tonoplast are energized by two proton pumps, a vacuolar H ATPase and H pyrophosphatase, means VPPase. First, we will discuss vacuolar cation channels. So, example is TPKs, TPKs. TPKs means two-pore potassium channels. They play a crucial role in regulating potassium transport between the vacuole and the cytoplasm, impacting various cellular processes. Uh, they are thought to act as sensors and participate in plant stress responses. They are also sensitive to changes in the osmotic pressure, potentially acting as osmosensor to trigger cellular response to osmotic stress. Then TPKs, particularly TPK1, are involved in voltage-dependent gating and are potential player in calcium-mediated signaling within the cell. Second type is TPC1 and SV channels. TPC1 or SV channels are slow activating non-selective cation channels primarily found in the vacuolar membrane of plant cells. They are encoded by TPC1 gene and are responsible for mediating flow of potassium and under certain conditions sodium across the vacuole membrane. So these channels play a crucial role in various cellular processes including stress responses and are implicated in the electrical excitability of the vacuolar membrane. This vacuolar cation channel is also involved in calcium-induced calcium release. Next is vacuolar anion channels. Examples are chloride channels, malate channels, putative SO42 and PO43 channels, calcium channels. So chloride channels involved in vacuolar chloride and this anion accumulation. Then malate channels, they are crucial for vacuolar malate accumulation, which is important for pH regulation and osmotic balance. Putative SO4 and SO42 and PO43 channels. They are involved in accumulation of sulfate and phosphate ions in the vacuole, and calcium channels play a role in the vacuolar ion transport and signaling. The slow vacuolar SV channels is a major cation channel, but it can also transport anions. Thank you.